Before we begin, I'd like to thank the James Family Charitable Foundation and the Al Held Foundation for making Art Science Connect and today's program possible. We've entered an era of do-it-yourself biology and biohacking. Who has access to that information? Who owns it? And what can they do with it? These biotechnology innovations raise fundamental questions about creativity and autonomy. Today's program showcases interdisciplinary scholarship that is both at the center and at the forefront of what we do in our graduate degree programs and research at the Graduate Center and the CUNY Advanced Science Research Center. As a chemist, a bioengineer, an arts advocate, and practitioner, I'm especially excited to welcome you to today's program.
One of the first works that I'm known for is Stranger Visions. This was my first transition into this field of biohacking. And this came really from the ground up from working in a community biology center called GenSpace, where I learned to extract DNA, analyze it, amplify it. And I used that to analyze forensic samples that I collected from the streets of New York City, things that strangers had left behind, like cigarette butts and chewed up gum and discarded hairs. And I analyzed those to create probabilistic portraits representing what they might look like based on their DNA information. T3511 is a short film, and this follows the story of a biohacker who falls in love with an anonymous saliva donor.
A cell is a history. A cell is a home. A cell is a whole. A cell is a cage. A cell is an electric fence, enclosing your legible unknown. To break the cell is to trespass the most intimate of spaces. Dear donor T2305, I received 5 milliliters of your saliva in June. It wasn't hard to get. Using my academic email and mailing address, I simply said it was for research, genetic testing, the truth. When the results finally trickled in 6 months later, I devoured your profile in a single sitting. You are 46 years old with dark brown eyes, a full head of brown hair, and few freckles. You toss and turn in bed, and you enjoy a savory midnight snack. You like to run, for miles, for hours, and this keeps you slim, keeps your heart beating strong, and helps tame your sometimes wild thoughts. And I wonder what you would think of me poring over your genetic details. I'd like to think you would be flattered, but you might be very angry. Dear donor, on Monday afternoon, I immortalized your cells. Your cells want to grow, and I passage them to feel you.
Today I will talk about the use of DNA and cells in a more commercial setting. Drug discovery starts with identification of a disease that doesn't have a treatment that's optimal at the moment yet. So there's people sick and there's no drug or no optimal drugs to treat the individuals that are suffering. Drug discovery process is basically split in 2 parts. Phase 1 is the preclinical phase, and phase 2 is the clinical trial. Preclinical phase takes roughly 3 to 6 years. So this goes from we have a disease and we have a lot of drug components that could potentially work, and then they test it one by one at whether indeed those work and indeed those cells respond well to it. The methods that are used now are computational models. So that's all based on a lot of data that's currently being used in order to extrapolate more information, but also cell models. So use human cell models for the screening of components, but also to understand how a disease comes about. Also how a drug affects a single cell, because if you understand the single cell, you can now extrapolate it also to a whole body. Only after this phase, and this has all been shown to work effectively to address that disease, you transit to the clinical trials to human individuals to test the drug onwards. The preclinical phase is really stooled on 3 important pillars. The DNA, so this is the blueprint of the individual that exhibits the disease. This is very important in order to understand this relationship, understanding that certain DNA codes for certain things that then being exhibited in a body or manifested in a body in a certain way. 23andMe has historically always focused on the profiling DNA from consumers, where indeed an individual can choose to send their DNA in order to get information about their familial relationship or their ancestry. But the DNA material also, they realized they could also, of course, use that for clinical applications. 23andMe now has 13 million, I think in the meantime already more, individuals in their database. But they had the DNA information, but they didn't have the bodily phenotypes associated with it. So my profile is in 23andMe. They write me an email asking like, hey, Sophie, we would like to ask you some questions about you. So for instance, I got the question, do you like the taste of dark licorice? Now, since I'm Dutch, I love dark licorice. And maybe they can be genetic. Maybe all Dutch people like dark licorice. They don't know, but they need from a lot of people their DNA profiles and whether indeed that's happening, and then they can correlate that together and they can link that together, and then they can at a certain point extract if there are certain loci in the genome that are responsible for this. Not only 23andMe, but also a lot in clinical settings. So a lot of hospitals, academic hospitals are doing this. They take samples from individuals and they link it to clinical manifestations of a specific disease. They also often take cell samples, they immortalize them and continue studying them in order to understand why this disease came as it was. You need a lot of individuals to understand the variation within the entire human population. So therefore, you equally need also the same variation in your preclinical work, DNA profiles, cell samples from individuals, in order to address that variation in the human population, in order to de-risk when you go from the laboratory setting to a clinical setting.
You know, I read this foundational piece by Nancy Shepard-Hughes and Margaret Locke, both medical anthropologists. And so they look at the body in three components, right? So the individual or physical, the second is social, the third is biopolitical. So after listening to your presentations today, I feel this is a really good place to start. Do you feel that there's another type of body that should be added?
You have to indeed consider social and cultural components of what people's beliefs are, also to how cells are being stored in a biobank. There are certain cultures that have religious components that they don't want their living material to live onwards outside of the body, and that is a complex matter. But that's something that I think for medicine is very important to address and discuss. As long as you open it up for discussion to understand each other's position, then you may be able to find a solution.
It's interesting to think about this question of if there's even something else missing, because the two are already not usually included. So, I mean, it's usually just the physical body basically that's considered, and the questions around biopolitics and the social side of things are given so much less consideration, even right now. But I really think the priority has to go to realizing first that there is this layer of ethical, of social, of political implications of doing this kind of work, and address that honestly. There was a review under the Obama administration of basically whether or not DNA used in medical research could be considered anonymous if it was de-identified. And the political lobbying weighed in so heavily that that was not considered. So even though the science strongly showed that you couldn't consider these samples to be anonymous properly, it was not taken seriously within the regulatory framework. It seems to me that that is at least an important precedent, an important building block, an important starting place that we have to acknowledge, first of all, that these things are people, that samples are people, that they have lives and implications and afterlives attached to them.
So when something is possible, when you're building to work towards something, it's not always obvious what the ethical implications are. Very famous example of Henrietta Lack's cells being the first immortalized cells that are in the Petri dish and are still cultured and used today, 70 years later. So 70 years she has been immortalized. But back at the end, so there's a lot of discussion about that she didn't give consent, which was absolutely a horrible situation for her and the whole family. But what I want to point out is that at that time, cell culture, also there were a lot of cell samples taken from individuals, and they tried to immortalize them because they knew it would be valuable to study an individual first in the petri dish before studying an individual as an entire individual. Then her cells were the first ones to grow, but they didn't realize that it would take off like that. They didn't realize the scope of what they created. And sometimes it's difficult. That's why I like the connection with the arts and the sciences, because I think art sometimes thinks more about the future. What are implications beyond what's possible now, in order to open up people's way of thinking, and in order to trigger, like, is it okay what's happening, what if this or what if that, and then you can start addressing it.
How do you deal with this DNA sequencing and drug trial information you are sharing in context of sexist and racist health disparities?
I think this is a critical problem in the biomedical field. The medical field is highly biased towards Northern European individuals. Cell samples are now 95% of them, and that's changing now very rapidly, but is of Northern European descent, which means that all the drugs are being screened and tested for Northern European individuals. And therefore, if you go to clinical trials, which is also, by the way, indeed often biased, it doesn't always translate. And there are very clear examples of drugs that are being developed solely stooled on Northern European individuals have adverse effects in, let's say, African individuals. It's a complex matter and it's changing, but I think there is a lot of push needed in order to really make this happen in a timely fashion.
Just to reiterate, there are really good reasons to have reservations about participating in all of these things. And so it's not like it's paranoia or conspiracy theory. And there's been many instances of really publicly known misuses. And beyond that, I mean, there is this kind of shady underworld of this afterlife of cells and data. Again, like the most basic thing we need is to have a real framework of consent, which means if I give you my saliva, let's say, that I know every use of that and that I'm asked permission before it's used in every instance afterward. I think that's really, it's a basic right that someone should have to have this kind of autonomy over what happens with their cells, with their data, this most personal of data that literally is giving rise to them, to their offspring, to their other relatives. So I think we need to have this basic framework in place where we can trust that we know what will happen.
This leads me to my question as to how biological data, such as 23andMe, are used in order to construct unsubstantiated anthropological, historical, nationalistic theses only with a superficial and obfuscated scientific patina. How dangerous is such a direction, and can scientists help put the proper limits of what science can and cannot say?
And we see this all the time. So basically that we have this conflation of all kinds of things with genetics. So conflation of race, conflation of ethnicities, conflation of religion, sexual orientation, intelligence. And you just go down the list and you can find any number of things that are socially constructed that are then read into and out of the genome. And the problem with data, when you have enough of it, you can usually find something there. And so this is part of what I've tried to do in my work, is to also show that many different interpretations are possible. You might be able to find a correlation between people, but it doesn't mean that that has caused anything. And it doesn't mean you wouldn't find also a correlation between different people. There's a lot of layers there to unpack, but I think in general, it is this kind of genetic reductionism and this wishing to find these social things within the genome that gives rise to this problem.
So people scratch the surface of, so how does it work? How does this DNA relate to us? So that DNA component with how does it relate in our population? We know a lot, but we also know very little. You have this whole migration, the evolution of the human species, if you will, that migrated, split off human populations. And that is what we are trying to understand now and try to put in segments in order to understand it better. We always try to understand things. So it's easiest to put it sometimes in a box in order to give it an easier way to understand it. But yeah, it's not easy, and it's indeed sometimes it's wrong.